Hello everyone. So we will continue in this chapter about the chromosome theory of inheritance. So up to this point, we learned that chromosome they can determine sex. Walter Sawton examined sex determination in a grasshopper, and he found that the type of sex chromosome is important to determine the sex of the grasshopper. So the Y chromosome, the presence of the Y chromosome determine maleness, the absence of Y chromosome determine femaleness. While Walter Fleming and all cell biologists, among of them is Boveri, he followed the behavior of a chromosome during mitosis and meiosis, and more importantly, during meiosis, the behavior of a chromosome is very similar to the behavior of alleles that Mendel suggested. So they came up with a theory. It's known as Sawton-Boveri chromosome theory of inheritance, suggesting that two copies of each kind of chromosome are there. So we have the homologous chromosome, two copies identical in shape, the same location of centromere band in pattern is the same, and they carry the same types of genes. And the chromosome complement, the total number of a chromosome, do not change from generation to another, meaning that in parent somatic cells in a human we have 46 chromosome, the same thing in the offspring somatic cells. So there is no gain or loss. This occurred normally. Homologous chromosomes separate to different gametes, they split, they move to different one, and during this division, the paternal and the maternal one, those chromosome, the homologous chromosome that they were originally came from the father and the mother, they move to opposite pole. And then during fertilization, then they reunite again. And if we look at any cells that they form after the fusion of the sperm and the egg, half of the chromosomes they will be from the father and half of the chromosomes will be coming from the mother. So this theory suggested by Boveri and Sawton uh, needs to be validated. It needs to be proven that it's correct. Up to this point, the only proofs came from sex determination, that type of a chromosome determines sex, and which is this, as we talked in the class, we said that this is a phenotype, so a gene on the Y chromosome determine sex. Sex is a phenotype, and another observation is the behavior of a chromosome. But for this theory to be true, it has to be validated using more example, meaning that we need to prove that there are other phenotypes other than sex determination are controlled by genes, and these genes are actually located in a specific chromosome. Now, if you want to see how the chromosome theory of inheritance verify Mendel first and second law, we look at these chromosomes, and let's say that this is the gene that determine either around the shape of the seed in peas round, and this one determine wrinkles. So these are located on homologous chromosomes. So these chromosomes are really similar in all aspects except that the type of allele here is different. So what will happen? Mendel suggested that these alleles they move away from each other, and now we know that these chromosomes during meiosis they move away from each other. So the gametes will have one copy either of the R capital or one copy of the R small. And in females, the same thing. In males and females, there will be one of two options, either the R capital allele or the R capital chromosome or the R small chromosome. So when they meet randomly in fertilization, then there will be an option of these the dominant allele to come together. Only one option out of four. Two options for the R capital R small one time the R small chromosome comes from the father, the R capital allele comes from the mother, and another way in which the R small comes from the mother and the R capital allele comes from the father. These are represented by two chances out of four. And the last chance, the last option is coming of two R small, the recessive allele from the father and mother, 1 out of 4. So by applying the chromosome theory of inheritance, Mendel's first law, the law of segregation, is validated. Does also the law of independent assortment also validated using the chromosome theory of inheritance? Let's look at this example. Remember now the round shape, R capital R small, and also at the same time we are looking at the color of the peas. Y capital determine yellow and Y small determine round. And let's say that the organism is double heterozygous, meaning that it has R capital R small, Y capital Y small. So one option is that when these chromosomes are arranged at the metaphase plate, the R capital allele and the Y capital allele will be in the same side, while the R small, Y small in the same side. This will generate two kinds of gametes. The other option, which is the same, has the same probability, is that the R capital allele will be segregating with the Y small allele, and the R small allele is segregating with the Y capital allele. This will produce also two kinds of gametes, Y capital R capital and Y Y small R capital, meaning that these four kinds of gametes are equally likely to be producing, that they have the same probability, meaning that each one of them can be produced by 25% chance. And then when they come together, they will produce 16 chances um, of genotypes, in which we studied them previously in the second. So now we will talk about the validation of the chromosome theory of inheritance. And as we said previously, before 1910, the proofs to the validation of the chromosome theory of inheritance comes from the sex determination that is associated with a particular chromosome by Sultan, and the evidence of mitosis-meiosis behavior of the chromosome and gametogenesis resemble the behavior of alleles that Mendel described. And this theory needs to be further validated. And here comes the importance of the experiments or the conclusions or the results of a very famous scientist. It's called Morgan. So Morgan used Drosophila melanogaster as a model organism, and I know a lot of you worked with Drosophila in the laboratory, so you know how they look. They are known as the fruit flies. So you can see them on ripened banana or a ripened uh, fruits. And the way how we write the genotype here, it's a little bit different. So the wild type of a gene, we write a plus, a superscript, and then a plus. And then the mutant allele, the same thing, but without a plus. So Morgan, he was looking at Drosophila melanogaster, and the wild type color of Drosophila is red. So he noticed that some flies, they had a mutant eye color, a different eye color, and it was white. So he named the gene the white gene here, the white gene. Why he called it white? Because the phenotype of the mutation is white. So he named the gene based on the phenotype of the mutant gene, not by the phenotype of the wild type. So the wild type allele determine red color, but the mutant allele determine white. And this is a recessive mutation. So you need two white, two W recessive alleles to see the phenotype. Similarly, so this is a recessive mutation, but if the mutation is dominant, the first letter of the mutant phenotype will be capital letter. So if you encounter the bar I shape, so the deviation of the allele is BAR and the B is capital, plus for the wild type and without a plus for the mutant. And by the way, this is a dominant mutation, meaning that inheriting only one copy of the bar in mutation is enough to cause bar I. So what Morgan noticed actually when he followed the behavior or the inheritance of this trait. First, the trait only appeared in males, so it was sex specific. Only males had initially the white phenotype. So let's follow four different types of crosses. At the end of those crosses, Morgan and his students concluded that white is X chromosome linked trait. So ignore the genotype here. We will come back to the way how we write the genotype when the gene is X-linked. So first time he saw a male, a Drosophila male with white eye, he crossed it to true breeding red females. So all the males and females of progeny, the F1, they were red eye. Okay, this is the first cross, cross A. Then in the cross B, he crossed those male and female siblings to each other. All right. So, and he got 3-to-1 ratio, 3 red to 1, 75% they were red, while only 25% a quarter they were white. This is very similar to what Mendel found in the monohybrid cross, except that in the exception here, that the mutant one, the Drosophila that they had the recessive phenotype, all of them were males. So half of the males they were red, half of the males were white, and 100% of females they were red. This is very interesting. So again here, the mutant phenotype, it was sex specific, only found in males. It appeared like that the females cannot be white, but it's not the case. So when he crossed a male, a white male, with the heterozygous female, now he got 1-to-1 ratio. So half of the progeny they were red, half of the progeny they were white, half of the females they were red, half of the females they were white, half of the males they were red, half of the males they were white. And the most important cross out of those four crosses is known as the crest cross, Huxley, in which if you have a true breeding females, white female, crossed to a red male, all of the daughters of this red male will be red, and all of the sons of this white female will be white. It's like the son inherit the phenotype of the mother, while the daughters inherit the phenotype of the father. So he brought all of the results of these crosses all together, and he went back and he analyzed these results, and he came up with very important conclusion. Let's go back to the crosses. It seems I can't go back from here. Oh, let's see. Let's stop the video and I will go back. All right, I couldn't go back to the previous slide because it already has a recording, so I could not override the recording on the top of it. But when he analyzed the results of the four crosses, he concluded that the mutation is X-linked. And just focusing at this cross number D, if the females are white, and because the mutation is recessive, they had to be homozygous. So X superscript W, X superscript W, while the males, because they are only having one copy of the X chromosome because their karyotype is XY and they are red, they have the wild type copy of the white gene. And now by following the segregation of the X chromosome, we know that we get as a males, we get the X chromosome from the mother and we get the Y chromosome from the father, right? So we have to inherit one of these X chromosome from the mother, and we can see that both of them here, they are mutants. So that's why all males are white. While in females, they have to inherit one X chromosome from the father. They cannot inherit the Y chromosome, otherwise they will become males. And this X chromosome has the wild type allele. And they will take one of those two X's, and these are the same. So the females will be heterozygous. But the males, they cannot, by the way, be what? Homozygous for X-linked traits. Males, we call them hemizygous for X-linked traits. They will have either the wild type copy of the allele, or can have the mutant copy of the allele. Another important experiment. So Morgan at that time, he was Morgan at that time, he was really excited about the results of the white gene, and he was excited to prove that indeed genes are carried on the chromosomes. But he was skeptical a little bit. He was not convinced 100%. So one of his students, Calvin Bridge, he repeated the crisscross experiment, but this time he did it on a large scale. Instead of starting with a few males and females, he did it with hundreds of males and females, so he could get progenies of thousands. So he repeated the cross. Wild type white females, they had two copies of the white allele, the mutant one, and males, they were hemizygous for the wild type. So he got most of the females, they were red as expected, and most of the males, they were white as expected. But he actually got some results that was really weird, that was really unexpected, in which he got some males that they were red and some females that they were white. And the only explanation about this is by non-disjunction. Then no segregation of paternal and paternal chromosomes from each other. So let's analyze how he could get only small fraction of males with with red color, while only 1,000 of females with white eye color. So let's look at the first scenario in which a non-disjunction occurs in female meiosis in the mother. So instead of these X chromosome to split to the daughter cells, they come together. So they will form oocytes with two X's and another oocyte with only one, with no X chromosome. So sorry. And if this egg is involved in fertilization with a sperm with the wild type allele, so three X's will be formed, and we knew from before that this is lethal in Drosophila. While if these two X's are involved with sperm coming from male with Y chromosome, they will form XXY. Is this a male or a female? Remember that Drosophila Y chromosome has nothing to do with sex determination. Sex determination depends on the number of X chromosome. So if we have two or more, it means females, and because this individual has two, it means that it's a female with a white phenotype because it has two white mutant alleles. So the only way how female progeny can be white is because of non-disjunction occurring in female meiosis. Then how can we explain how males that are supposed to be white because they has to inherit X chromosome from the mother, how they could be red? So similarly, in the same non-disjunction, uh, remember that the two X's they were segregating to one egg. The other one is empty. It has no X chromosome. So if that one is involved with the fertilization with X chromosome come from a male, it will form what? It will form an individual with one X chromosome, and this one will be a male, although it has no Y chromosome, but Y chromosome again has nothing to do with sex determination, and this male will be red. And this is the only way how non-disjunction will deviate the expected results from normal segregation of the chromosome during meiosis. But this is very important because it also proves that the chromosome theory of inheritance is correct. This is the end of this part, this lecture, and I will see you in another lecture soon, inshallah.